Hey everyone, welcome back to Guys Teach Stuff. So today you will be learning about cells and microscopes, topic 7AC and 7AD in your Exploring Science textbook 7 with myself, Aditya. So let's first take a look at cells. So what exactly is a cell? Well, a cell is a mass of cytoplasm that is bound externally by a cell membrane. I know that sounds quite confusing, but it is actually quite simple. Normally, cells are the smallest structural units of living matter, and it is what most living things are made out of. To put it into perspective, in the human body, there are somewhat 37.2 trillion cells. I know, that sounds like a lot, and well, to be frank, it is. And you may be wondering, how does it all fit in our body? The answer is that they are so very tiny that all of them can fit inside of our body. Let's look specifically in an animal cell. The photograph I'm showing you has a magnification of x700, which means that it is 700 times larger in this photo than it is in real life. Now you may understand how it fits in our body. In the photo, you have the cell surface membrane, which is a very thin bag. Think of it as a plastic bag, or even thinner. It keeps the cell together, and it controls what enters and leaves the cell. It's kind of like the security guard of the cell. It controls what comes in and out of the cell and secures the shape and position of the cell. The nucleus is the brains of the cell. It controls the cell and the activity that goes on within it. It is very dense, meaning that the size is extremely small, but it has a very high mass, as most of the cell's weight is concentrated into the nucleus. The cytoplasm is practically a watery jelly. You know when you tried to make slime for maybe the first time, but it failed and was way too watery? It's basically that. I know, it's quite disgusting. Most of the cell's activity, however, happens here, and this includes respiration, which happens in tiny structures called mitochondria, which I'll explain later, which have a jelly bean shape. All of the animal cells have the same basic parts, but many of the cells from different tissues have different sizes, shapes, and functions to help them do their jobs. These cells are given the term specialized. An example of a specialized cells are heart muscle cells, which work together in heart muscle tissues. They're specialized to make the heart move. Another example is a root hair cell. These cells are specialized to take water from the soil. Now that we're done with animal cells, let's take a look at plant cells. Plant cells have thick cell walls and many other features which animal cells do not have. I will explain the different new structures that are not in the animal cells instead of repeating what was in the animal cells. So, first of all, plant cells contain chloroplasts. These are green discs, which also have a somewhat jelly bean shape. They are in the cytoplasm and they contain chlorophyll. The chloroplasts make food for the plant using photosynthesis, which will be covered in a future video. Secondly, plant cells contain vacuoles, which are also known as large vacuoles. The vacuole is a large storage space, and it is filled with cell sap. Third of all, and finally, plant cells contain a cell wall. This is a kind of box with many holes in it, and it supports the cell. It is made of cellulose, which is extremely strong. Think of it as an upgraded cell surface membrane. However, the plant cell still contains a nucleus, cytoplasm, and a cell surface membrane. Now, you may be wondering, Aditya, how do we even see the cell? And how do we even know what is in the cell? Well, that is the job of a light microscope. So, how is a light microscope used to examine a specimen? Let me picture this for you. You go to the doctor and there's something wrong with one of your organs. It's pretty common. To find out what is wrong with the organ, the doctors do a series of tests. Some of these tests involve taking small pieces of tissue, which is called a biopsy, from an organ and they look at it under a light microscope. Microscopes make things appear bigger, as their jobs, if you haven't already guessed it, is to enlarge or magnify things. Now, let me explain the steps on how to use a light microscope. Part 1. Place a small objective lens, the part that does the enlarging and the magnification, and the smallest one, over the hole in the stage. Next, turn the coarse focusing wheel to make the gap between the objective lens and the stage as small as possible. Part 2. Place a slide under the clips on the stage. The slide contains the specimen, the thing you want to look at. Then adjust the light source so that the light goes up through the hole. Part 3. Look through the eyepiece lens. Turn the coarse focusing wheel slowly until you see it is in focus, which basically means that it is clean and sharp. Part 4. To see the image bigger, place a larger objective lens over your specimen. And finally, which is part 5, use the fine focusing wheel to get your image in focus again. Do not use a coarse focusing wheel, which you should have been using, since you can break the slide and damage the objective lens. And trust me, from experience, this will not make your teachers happy. If you cannot see your specimen clear enough, then go back to the lower magnification. However, I do need to warn you guys. Please, for the love of God, never point a microscope mirror at the sun unless you want permanent damage to your eyesight. If you do want to go blind, however, then go for it. I'm not going to stop you. Now, I know there is some maths involved. On this side of the slide will be written the magnification. To work out the total magnification, use the formula magnification of eyepiece lens times magnification of objective lens equals total magnification. Now, we need to know how to prepare a specimen. The specimen needs to be thin so that light can pass through it. Let me explain the method. Step 1. Take a slide and place a drop of water in the center. The water may contain a stain to make the specimen show up better. Step 2. Use some forceps to peel off the inside layer of a piece of onion, for example. Step 3. Place your onion skin into the drop of water on your slide. Step 4. Use some forceps to lower a cover slip, which is a thin glass object, to cover your specimen. If you do this carefully and slowly, you will not get any air bubbles trapped underneath the cover slip. Now I've explained everything, let's look at this checklist. You should be able to identify the main parts of animal cells and plant cells, and you should also be able to describe their functions. Additionally, you should be able to describe how to prepare a microscope slide and how to describe how to use a light microscope to examine a specimen. Thank you very much for watching Guys Teach Stuff. Please come back to this channel for more science videos that will be uploaded in the near future. Thank you, that's it for me.